Welcome to the Proteomics in Proximity podcast, where your co-hosts Dale Yuzuki, Cindy Lawley and Sarantis Clamidus from Olink Proteomics talk about the intersection of proteomics with genomics for drug target discovery, the application of proteomics to reveal disease biomarkers, and current trends in using proteomics to unlock biological mechanisms. Here we have your hosts, Dale, Cindy and Sarantis.
Thank you for joining us on Proteomics in Proximity. I'm your host, Dale Yuzuki, with my co-host, Cindy Lawley, and our co-host, Sarantis Clamidus. This afternoon, we're going to go ahead and talk about BRCA2 crisis. Sarantis, go right ahead.
Thank you, Dale. Thank you, Cindy. Yeah, today I choose a paper from Snyder's lab where it describes the mechanism of action in the cancer progression of the BRCA2 mutations. And I found it really interesting, this paper, because it goes to the details of molecular mechanism, how these mutations regulates, let's say, cancer progression. We know BRCA2 is a famous target for breast cancer and ovarian cancer. We know that when it's mutated, PARP proteins, they play the role of DNA repairing. And there are a lot of PARP inhibitors now also FDA approved that for therapies, for cancer therapies.
Now, if I remember correctly, Sarantis, BRCA was involved in this sort of DNA repair, right? Non-homologous DNA end repair, the sort of keeping the integrity of the genome. And when people have, particularly women, have mutations in the BRCA genes, they're susceptible just because their DNA gets screwed up. Is that basically it?
And it's men and women.
Right.
Yeah.
And actually, recruits all the repair machinery interacts with RAT51, where it's a recombinase for the homologous recombination repairing. But also has been shown recently, quite recently, with a lot of papers that helps of the dissolving of R loops. And R loops, as you may know, is hybrids of RNA DNA. We have it in our genome during replications, but also during transcription. And when we have accumulation of R loops, we have a lot of genome instability and DNA breakages. Our BRCA function is actually to dissolve this and maintain genome integrity. On top of this, by dissolving this R loop formation helps POL2 progression, because this POL2, with this way, POL2 can travel really nicely along the gene, and we have expression. That means it has a multifunctional role in the genome by regulating gene expression directly, but also interfere with the R loop formation and genome stability. It's a great protein, actually.
Wow. And this particular paper you're pointing to in Cell Reports, this is from Mike Snyder's group, right?
Yes.
Chromatin remodeling in response to BRCA2 crisis. So what exactly was
Josh Gruber. Josh Gruber and Mike Snyder. Yeah, 2019.
From 2019. And what was the problem they were trying to solve?
Actually, they wanted to see what is the situation, what are the epigenetic mechanisms together with a genetic mutation that create carcinogenesis. They use an in vitro model by transit depleting BRCA2, and this was really clever tool, because so far a knockout mice or a permanent depletion of BRCA2 creates cell arrest and death, and it's not easy to study under these conditions, obviously, the effects of the protein. They created transient transfection, transient depletion of the BRCA2, and then let the cells recover. And by this, they can see which epigenetic marks they can be deposited and which genes are regulated by this, let's say, epigenetic mechanism through BRCA.
So changes in expression of those genes.
Changes in expression, changes in epigenetics, and by having the cells growing in the EGF-free medium, they have an assay for carcinogenesis, because, as you know, cancer creates proliferation, and this assay, by showing overproliferations in BRCA2 crisis cells, they were able to assess carcinogenesis, the early carcinogenesis steps in these cells.
And you mentioned EGF-free medium, but becomes important due to the dependency, is that correct? Dependency of cancer cells for EGF and EGFR receptor pathway?
Yes. Actually, normal cells, they need the EGF also to grow, right? But the cancer cells, without the EGF, they can overgrow and they can overpass this, and that was a model to assess carcinogenesis in this vitro system that they have generated. And as you can imagine, BRCA2 creates carcinogenesis through epigenetic mechanisms.
And what kind of epigenetic mechanisms did they go ahead and take a look at?
Yeah, it's really nice because they check different angles. They check that they first look at DNA methylation. And DNA methylation per se, it didn't create so much difference when they mutate BRCA2. Then they go with the ATAC-seq. And ATAC-seq is an assay that originally created by Greenleaf's lab, where with this assay, we can measure differential accessibility of the chromatin. In few words, there's transposase based, where you have barcodes. By activating the transposase, we have integration of the barcodes in the open chromatin region and sequence the open chromatin region. That means we can assess which regions get differentially open upon different treatments. In that case, BRCA2 depletion.
So if I understand ATAC-seq, you mentioned that the methylation status didn't give you differential methylation, but ATAC-seq showed you which was open. Is that histone dependent then?
Exactly. This could be both. Could be histone dependent, could be non-coding RNA dependent, could be transcription factor dependent, right? And then the next step of their study was to understand if there were some connection with the histone marks. As you know, histone acetylation is connected to the opening of the chromatin. They check a series of histone acetylations. Here, parenthesis, there were some studies showing that BRCA2 has acetyltransferase activity also at the level of histones. It's really amazing what this protein does.
It's
actually
a complex system. Yeah.
Absolutely.
Getting at the functional genomics here is amazing.
To repeat, you said that BRCA has histone acetylation activity.
Yeah, okay. There are a paper, Nokogene, I think, that they have seen that has acetyltransferase activity, or is connected to, let's say, histone acetylation somehow. It's crazy. Yeah, it's really crazy. And by having Western blots different histone with different histone marks, they see that most affected is H4K5 acetylation and H4K12 acetylation. Actually, by depleting BRCA2, the levels of these marks in EGF-free medium, they get in high. And you have actually hypertranscription of certain genes. But with RNA-seq...
Yeah, which we can imagine would manifest, hopefully, logically manifest as protein differences in those tissues, yeah.
Absolutely. And then they check RNA-seq levels, and they see nice correlation with BRCA2 binding by ChIP-seq, H4K12 acetylation, and expression of the gene. That means that BRCA2 through H4K12 acetylation and H4K5 acetylation regulates gene expression. And when you have depletion of BRCA2, we have misregulation of certain genes. A lot of these genes, I have to say, that they are connected, as you can imagine, to cell cycle, to signaling, and of course, by misregulating with this gene, you enhance and you create cancer at the end, right? That's the thing.
So as far as then this global gene expression, was it hundreds? Was it thousands of transcripts?
Yeah.
Of different types of genes?
Yeah, they were initially they see with RNA-seq, I think they were thousands of genes, 3,000 or that, misregulated of that.
Yes.
On top of this, there's also a crosstalk with NF-kappa-B. They see that the depletion of BRCA2 leads to NF-kappa-B activation. And,
now, help me. Maybe it's been too long a time since I've studied immunology, right? NF-kappa-B is a key molecule.
It's a cortical, regulates the expression of cytokinase, among other things. Yeah, and when it's activated, it's translocated to the nucleus, binding to a key response genes, interleukins, chemokines, and of course is a master regulator, let's say, if we can say about that. And they have seen that NF-kappa B is upregulated. We have overtranscription of cytokinase, but nicely, using proteomics approach, they close the circle and they see that not only you have a hypertranscription of cytokinase, but also secretion of the cytokinase in the cell.
Hang on. So we're not talking about immune cells. We're talking about human breast epithelial cells.
Non-transformed epithelial cells.
Yes. So this is a BRCA2 induced situation, kind of like a loss of heterozygosity situation. We have a single copy of BRCA2 gene that was sort of induced. And what you're telling me is that BRCA2 is affecting, is it the K12 acetylation?
K12 and K5.
Okay, K5 acetylation that turns on NF-kappa B. That then turns on cytokine production?
Cytokine production, yeah. And they see it by screening like a panel of 92 cytokines in the media of the cells, and they were able to see that 7 of them, they are hyper-present, hyper-transcribed, and hyper-produced. And it was really nice phenotype. It was really nice phenotypically to show the effect of BRCA2 on the cytokine production.
Now, why would a mammalian breast cell produce cytokines? I mean, maybe I'm stuck on that as far as activating the immune system.
I think this is coming through NF-kappa B, right? This is coming through NF-kappa B, actually. And I'm guessing because BRCA2 creates a lot of dysregulation of genes, but also I could imagine that you have some DNA damage also that is happening there, and they haven't assessed that. They haven't gone on that. They haven't really checked in the details which type of damage they may have created that also create a stimulus to production of cytokinase. Right? I'm guessing.
And how do they determine it was NF-kappa B that was turned on? Was that again through RNA data?
Yeah, they see that the genes that are misregulated with BRCA2, a lot of them has been reported that they're regulated by NF-kappa B from previous studies that they have done. And then they just went to see what is the situation with NF-kappa B. That was the.
I see.
That was the.
And that was where Olink came in, was that they were able to measure the cytokines just from the cell supernatant.
Exactly. And so phenotypically, the effect on the cell and how this may interfere with cell progression, cancer progression, and potential metastasis in the future, right?
Yeah. And then I guess the bottom line then, as far as discovering this particular model, how can that be then further applied to potential treatment or perhaps a diagnostic? I mean, is that...
I think, first, I think we have to say here that they didn't stay only to this in vitro model. They check also in vivo, and they see for patient samples, they have checked that they have exactly the same effects at the level of gene expression, and the genes that they are highly expressed in mutant patient, they are the same in the BRCA crisis cells. That means, and there is also increase of H4, K5, and K12 acetylation on these genes. That's the mechanism that they see also in vivo. And really nicely also, they make a xenograph model, and they can see that BRCA2 crisis cells growth in HGF-free medium, they have bigger tumors. And really metastatic, they see really metastatic and tumor growth effect at the end.
Sarantis, is there...
Okay.
Oh, I was just going to ask, is there a proposed mechanism by which this might get turned on, right? Because presumably the genetic state of BRCA someone has through their lifetime, at some point, for some reason, carcinogenesis is started. There's this, lack of dissolving of this component. And I'm just wondering if you have any thoughts, and I don't think they mentioned it in the paper, what might turn on such a mechanism?
I think it's a difficult question. Definitely, for sure. Definitely, I think there's more than one mechanism here. I don't think that it's only H4K12 acetylation. I don't think that's only chromatin related. I think it's also POL2 as I mentioned before, hybrids, R-loop formation, TIP60. I think there's a set of information and a set of mechanisms that may interfere, but it was really surprising and astonishing the crosstalk with NF-kappa B and the acetylation of the histones. And I think this is the novelty that they bring in this aspect. And coming back to your question, Dale, in regards how we can use this information for prognosis or for treatment, I think knowing now that there are a lot of mechanisms epigenetic that interfere with BRCA2, I think we can combine in the future drugs that are, for example, against DNA methylation or against acetylations, right? And we can have combined cancer treatments, for example. Give us a little bit of new ways of thinking of how we can target these mechanisms.
Yeah. The challenge, right, is epigenetic modifiers. There's only a handful of them on market, if I understand, in terms of druggable targets. Do you have any comment on that in terms of drugs that target sort of epigenetic effects for health rather than sort of other conventional drug targets?
I think as far as I know, there are very few drugs FDA approved. They're epigenetic drugs. I know that the field is expanding and there are a lot of works out there, but people they're working on that. One of the problems is that these enzymes, they don't have only single targets. Like they can either modify histones, but also can modify proteins, right?
Yeah, delivery to the right place.
Yeah. Exactly. And I'm guessing that they have a lot of toxicity effects that they may have to
mediate.
Yeah, exactly. That's one of my thoughts on that. But definitely, I know that it's an active work, and a lot of people they are looking on that. And I hope things will get better in the future with more small molecules inhibitors and against the enzymes, epigenetic enzymes.
Okay. Well, thank you, Sarantis, for sharing your thoughts today.
Thank you very much.
And being willing to speculate on, some of the aspects that weren't covered in the paper, right? Because this is it's such a fascinating area where we've had advances in diagnostics, certainly advances in treatment, and genetics has played some part in that, of course. But to really understand the mechanism, a clever system like this is amazing.
Insights.
To get at functional biology.
Right, yeah, to overlay RNA-seq data and then look at open chromatin, looking at histone modifications, and then getting down to network effects around NF-kappa B and exactly right where the cytokines are playing a role. Yeah. That aspect of multi-omics, I mean, it's here to stay. The very fact that these tools are available.
And here's to Mike Snyder's team for spearheading multi-omics in a way that few others have as early as his team has done this. This is, intense, incredible.
Super. Well, until next time, take care.
Thank you. Thank you very much.
Okay. All right.
Nice job, Sarantis.
Thank you for listening to the Proteomics in Proximity podcast, brought to you by Olink Proteomics. To contact the hosts or for further information, simply email info@olink.com.